Welcome back, ladies and gentlemen. So now that we have a messenger RNA molecule that has been processed and edited, now what? Well, that messenger RNA is now going to leave the nucleus, go out into the cytoplasm, and be ready for translation. Be ready for making that code and translating it into amino acids that we can actually build into proteins. But before we get there, there's going to be a couple of other players that come in this step. So let's start there and just make sure that we are all on the same page as to what molecules also come about when we talk about translation. And we're going to start here with transfer RNA. So we've got that messenger RNA, right, what we copied from the DNA code, but there are two other RNA molecules that also come in to the game when we get to translation. And this tRNA is your transfer RNA. And this is going to help to translate that messenger RNA into the proteins because these guys are the molecules that are hanging out in your cytoplasm, picking up amino acids, and then taking them to the ribosome themselves. So when we look at the structure, this is what a general tRNA will look like. It actually hairpins back on itself hydrogen bonds to make literally a T structure, which is sort of convenient in terms of the way that it looks. And then it is also acting as a transfer RNA, which is where the other T comes into it as well. So there's two really important attachment sites here when we look at tRNAs, and that's their amino acid attachment site and their anticodon attachment site. So those two codons they correspond to each other. So the amino acid attachment site is going to allow the attachment of a very specific amino acid, and that specific amino acid that it binds to is also very specific to the anticodon sequence that it has. Now that anticodon sequence is then going to complementary base pair to the sequence in the mRNA. So when we look at this, so if this was my anticodon sequence in my tRNA, then its complementary sequence would be the UUC in my messenger RNA, and then that also corresponds to the amino acid that it carries. So of a single RNA strand, there's about 80 nucleotides, give or take, that exist here. But again, the ones that are most important for us are for that attachment site and then the anticodon that then matches the messenger RNA molecule. So when we look at these tRNAs, in order for them to attach those amino acids and then ultimately carry them to the ribosome to match that messenger RNA sequence and then start building the protein, I've got to get the amino acid attached to that tRNA in the first place. And this is going to happen with the help of an enzyme called amino acetyl tRNA synthetase. So its job is to have a binding site in its active site for the amino acid. We're going to use some ATP to help facilitate this, but because this is an enzyme, I need less energy than I would if this enzyme didn't exist. We're also going to then fit in the tRNA, and we're going to be able to charge, basically, is what they call that, charge the tRNA with its corresponding amino acid. So in order for this to work, though, we have to correctly match the tRNA anticodon um, to the binding site to the correct amino acid. And then once that charged tRNA has been removed from this synthetase enzyme, it can then go complementary base pair with the messenger RNA at the ribosome. Now there is flexible base pairing here, which is pretty nice and pretty interesting in the way that it works. So what we mean by that is this idea of this wobble. So it's going to allow some tRNAs to bind to more than one messenger RNA codon. So when we start digging into that codon chart, right, there are going to be multiple codes, multiple mRNA codes that correspond to multiple tRNA codes, anticodons, because we have more than one code for an amino acid, which is good. So if some minor mistakes or minor changes happen to the DNA code, happen to the messenger RNA code, we don't ultimately immediately change the amino acid that gets coded for, and so the protein doesn't know the difference. So another RNA that comes into play with translation is ribosomal RNA. And that ribosomal RNA gets fitted along with a bunch of proteins to make your ribosomes themselves. And this is going to ultimately really be the site of that translation process. So when we look at ribosomes, remember that ribosomal RNA first and foremost gets made in the nucleolus of the nucleus and then gets exported, and we couple that with these proteins and make these ribosomes into two subunits. You've got your large subunit on the top and your small subunit in the bottom. And then there are binding sites within those subunits that will sandwich the messenger RNA within in order to read it appropriately, read it in the right order, and also have binding sites to start building that growing polypeptide. So I kind of like to think of ribosomes kind of like building your sandwich, right? You've got your large subunit at the top, which is the big sort of domed piece to your bun. You've got your messenger RNA fitted in between, where we're reading the codes, so sort of like the meat and lettuce and cheese on your sandwich, and then you've got the small subunit at the bottom, which would be the flatter part of that roll. So those binding sites each have their own corresponding function. When we look at your P site, this is the site here in the middle, and that's where most of the work is happening. This is where we're actually using the corresponding tRNA matching to the messenger RNA's codon, and we're starting to build the polypeptide chain. The A site is the attachment site. That's where the actual complementary sequencing is happening. So while I've got that messenger RNA in here, I've got that blank messenger RNA codon going on, and in and out are going to pop these tRNAs until I get a complementary match between the anticodon on a tRNA and the codon on the mRNA. The E site is where I'm going to kick out basically the uncharged tRNA. So once we get the match to happen in the A site, that all shifts over into the P site to start adding the amino acids. And then we shift this again one more time to that E site, where now that that tRNA no longer has the amino acid attached to it, it gets removed from the ribosome, goes back out into the cytoplasm, uses that synthetase enzyme to help charge it back up and attach the corresponding amino acid so that it can continue to help in the growing uh, protein process. So when we look at translation as a whole, we're going to break it down into 3 stages, just like we did with the transcription. And in fact, we're going to use the same terminology: the initiation, the elongation, and the termination. Now, all 3 of these stages are going to require protein factors that aid in the translation process to either keep things in place, move things along, so on and so forth. So let's dig into these processes. Okay, so initiation really is going to happen in that A site initially, and we're going to actually really build sort of this ribosomal sandwich. So I need to bring together the messenger RNA, that initial tRNA with the first amino acid, which, oh, by the way, will always be methionine and will always be that AUG sequence on my messenger RNA. And then once that is all fitted together, we're going to fit that complementary tRNA, messenger RNA with its corresponding amino acid. All of that ends up on the bottom subunit of that ribosome, and then we fit the top large subunit on top and make this initiation complex. All of this is being aided by initiation factors, so more proteins that help to bring in that large subunit, complete that initiation complex, and just kind of hold everything together, sandwich it all together, make sure that it stays together to keep the process moving. Elongation then is going to be the complementary base pairing in that A site, so finding the corresponding anticodon to the codon that is currently sitting in that A site. tRNAs will bounce in and out of that site until you find the match. Once that match is found, Then we're going to shuffle this over. So as this happens, we're going to use some GTP. So kind of think of it like ATP. It's an energy molecule, but it's a guanine instead of an adenine. And that's going to help facilitate the bonding of the growing polypeptide chain to the new amino acid that that tRNA is bringing in. That's going to shift everything over. And once that is separated, the now uncharged or blank tRNA moves into that E site, gets kicked out, and we move everything back over to keep growing. And this cycle continues to happen until we get all of the codons read. So we're going through this sort of codon recognition, which is what's happening here, the peptide bond formation that's happening here, and then the translocation idea here where we're translocating, we're moving those tRNAs into the next site to make that A site open again so that the codon recognition can happen again, peptide formation can continue to happen, and the translocation again continues. Cool thing about translation is that we can actually attach numerous ribosomes to the same messenger RNA. So once we've built that initial initiation complex onto that messenger RNA and we start going through elongation and reading it, we can then add another another ribosome onto that messenger RNA and keep the process going. So you get these polyribosomes or polysomes, and this is going to once again allow cells to increase the product that they're producing without needing to waste additional time and material making copious amounts of messenger RNAs. We can also add multiple ribosomes and make the protein, multiple copies of the protein from the same messenger RNA molecule. So again, we're reading this genetic code, right? How do we know how to build this? Well, it's a triplet code. So if I go back to that template strand of DNA, we make the copy of messenger RNA from there, and then when we get to the ribosomes, we're reading that messenger RNA 3 bases at a time. Every codon is 3 bases, which corresponds to 3 bases on an anticodon, and all of that gets translated into a single amino acid. So that template strand provides the original instructions that then ultimately get read from that messenger RNA in those codons. The other idea here is we have to remember we have to read this in the correct order. So it has to go from 5' to 3', so that way we are building the amino acids in the correct sequence. So when we look at the template strand of DNA, it's always the 3' to 5' strand that we read in order to make that 5' to 3' messenger RNA in the correct order. Now, there are 20 different amino acids, which means there has to be at least 20 different codons, 20 different anticodons in order to have a unique code for every amino acid. Now, I mentioned that idea of that wobble protein before, and that's because that wobble principle is going to become important when we look at the codes. There are actually more than 20. In fact, there are 64 of them. We actually figured this out back in the 1960s. So there are 61 codes specific for those 20 different amino acids, which means there are multiple codes for the same amino acid. And if you look at them, something like lysine here, the first 2 letters are consistent, and then that third letter is where that wobble comes in, where I can have any one of the 4 bases in that last code, and it still codes for the same amino acid, which means it doesn't ultimately change the protein. There are also 3 stop codons to be able to tell us this is the end of translation, and that AUG is always your start. All right, now these codons have to be read in the correct reading frame, meaning the right 3 bases at a time. If I start screwing up that reading frame, then we can start talking mutations, and that can have significant impacts on what amino acid gets brought in, and as a result, the protein folding at the end of that production of that polypeptide chain, which then has implications on whether or not that protein actually functions and does its job.
In order for our bodies to function, we need to supply them with a variety of nutrients we get from our diet. Our bodies cannot use the food as it is when it enters our digestive system. The process of chemical digestion uses different proteins and enzymes to break down the food particles into usable nutrients our cells can absorb. And where are the instructions to manufacture these and all the different types of proteins we need to stay alive? The instructions to make proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. For some genes, the encoded RNA is used to synthesize a protein in a process called gene expression. For these genes, expression can be divided into two processes: transcription and translation. In eukaryotic cells, transcription occurs in the nucleus, where DNA is used as a template to make messenger RNA. Then in translation, which occurs in the cytoplasm of the cell, the information contained in the messenger RNA is used to make a polypeptide. During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly-A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex removes the intron segments and joins the adjacent exons to produce a mature messenger RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3 letter codes called codons. The genetic code includes 64 codons. Most codons code for specific amino acids. There are 4 special codons, 1 that codes for start and 3 that code for stop. Translation begins with the messenger RNA strand binding to the small ribosomal subunit upstream of the start codon. Each amino acid is brought to the ribosome by a specific transfer RNA molecule. The type of amino acid is determined by the anticodon sequence of the transfer RNA. Complementary base pairing occurs between the codon of the messenger RNA and the anticodon of the transfer RNA. After the initiator transfer RNA molecule binds to the start codon, the large ribosomal subunit binds to form the translation complex, and initiation is complete. In the large ribosomal subunit, there are 3 distinct regions called the E, P, and A sites. During elongation, individual amino acids are brought to the messenger RNA strand by a transfer RNA molecule through complementary base pairing of the codons and anticodons. Each anticodon of a transfer RNA molecule corresponds to a particular amino acid. A charged transfer RNA molecule binds to the A site, and a peptide bond forms between its amino acid and the one attached to the transfer RNA molecule at the P site. The complex slides down one codon to the right, where the now uncharged transfer RNA molecule exits from the E site, and the A site is open to accept the next transfer RNA molecule. Elongation will continue until a stop codon is reached. A release factor binds to the A site at a stop codon, and the polypeptide is released from the transfer RNA in the P site. The entire complex dissociates and can reassemble to begin the process again at initiation. The purpose of translation is to produce polypeptides quickly and accurately. After dissociation, the polypeptide may need to be modified before it is ready to function. Modifications take place in different organelles for different proteins. In order for a digestive enzyme to be secreted into the stomach or intestines, the polypeptide is translated into the endoplasmic reticulum, modified as it passes through the Golgi, then secreted using a vesicle through the plasma membrane of the cell into the lumen of the digestive tract. Proteins are needed for most physiological functions of the body to occur properly, such as breaking down food particles in digestion, and the processes of transcription and translation make the production of proteins possible.
So when we look at gene expression among different domains of life, the coolest thing about this is that every organism on Earth, for the most part, seems to have a universal code. So while bacteria and eukarya may differ in their actual RNA polymerases in terms of the termination of transcripts, the way that the ribosomes are structured completely, the general principle works the same. So a messenger RNA in a bacteria works the same way as messenger RNA in any eukaryotic cell. Now, we do see that archaea, although they are prokaryotes, their polymerases and their true ribosome structure does look more similar to eukaryotes. Bacteria can simultaneously transcribe and translate genes at the same time. We can't do that in eukaryotic cells because of that nuclear envelope. We've got some of that extra processing that happens to the messenger RNA in a eukaryotic cell. But again, the general idea of the way this process works is consistent. There are some extra components to the steps, some minor differences in the molecules that facilitate this, but generally speaking, everything works the same. So we have this idea of a gene sort of as a unifying concept of life as a whole. A gene, period. Doesn't matter if I'm talking prokaryote, eukaryote, anywhere in between. A gene is a discrete unit of inheritance. It's a region that specifies a sequence in a chromosome, and then that sequence codes for a specific polypeptide chain. So the genetic code's universal, or at least nearly universal. It's shared by the most complex organisms down to the simplest ones. And so genes can be transcribed and translated after being transplanted from one species to another. This is really going to be the hallmark of why biotechnology is a thing and why it works. So I can take a firefly gene out of a firefly genome, stick it into a tobacco plant, and make it glow. I can do the same thing to a pig, take a bioluminescent jellyfish gene, put it into a pig, and make a glowing pig. That's pretty cool. Okay, so if the mechanism for expressing those genes is universal, how do I ultimately terminate the expression? Well, once I get to a stop codon, right, and there's 3 of them in your messenger RNA molecules, that's going to bring in this thing called a release factor. And that release factor is sort of like hitting the eject button or the destruct button. to this entire ribosomal complex. So once that release factor gets attached to the A site of the ribosome, it's going to bring in the addition of water molecules instead of an amino acid. And that reaction then releases the polypeptide chain from the P site, releases that final blank tRNA, and releases the mRNA from that ribosome. Everything basically falls apart. And that is then the end of that translation for that particular polypeptide chain.
Okay, roll it. You know what this is? It is the longest word in the world. Like anywhere, any language, more than 189,000 letters. If you were to write it down, though I don't know why you would, it would fill up more than 100 pages, and if you could actually say it without like breaking your face, it'd take about 5 hours. What the frick is this word? Well, it is the name of the longest known protein on Earth, and it's actually in you right now. Because of its enormous size, it was given the name Titan by scientists, and that's with 2 Y's. And it's a protein that helps give some like the springiness to your muscles. Today we're going to be talking about DNA and how it, along with 3 versions of its cousin RNA, unleash chemical kung fu to synthesize proteins just like this. So this is going to take a while to explain. So how about we make ourselves some Hot Pockets? Of my favorite, ham and cheese. Every time I take a bite, I wonder, how do they do it? How do they pack exactly the same flavor into every foil cardboard wrapped food-ish item? Clearly there's got to be some super secret instruction manual kept and a location known only to 2 people. And since I'm talking about biology here, that brings up a related question. How did I get built from the DNA instructions and biological molecules we've been talking about? Today, that's what I'm going to do. Not actually make Hot Pockets or a person, but I'm going to be talking about DNA transcription and translation, which is how we get made into the delicious things that we are today, though hopefully none of us know how delicious people are. Animals, plants, and also Hot Pockets really are nothing more than salty water, carbohydrates, fats, and proteins combined in precise proportions following very explicit instructions. Let's say I want to make my own Hot Pocket. I would have to, 1, break into the lair of the Hot Pocket company holding the secret manual, 2, read the instructions on how to make the machinery to produce the Hot Pocket and the proportions of the ingredients. 3, quickly write down that information in shorthand before I get caught by the Hot Pocket police. 4, go home, follow the instructions, build the machinery, and mix the ingredients together until I have a perfect Hot Pocket. And that is how we get us. Very simply, inside the cell's nucleus, the DNA instruction manual was copied gene by transcription onto a kind of RNA and then taken out of the layer where the instructions are followed by the process of translation to assemble amino acid strings into polypeptides or proteins that make up all kinds of stuff from this Titan down here to the keratin in my hair. But most of the polypeptides that get made aren't structural proteins like hair, they're enzymes which go on to act like the assembly machinery, breaking down and building and combining carbohydrates and lipids and proteins that make up variations of cell material. So enzymes are just like whatever ingenious machinery they use the factory to make this. Okay, let's start out in the layer. The nucleus. The length of DNA that we're going to be transcribing onto an RNA molecule is called our transcription unit. And let's say, in today's example, that it's going to include the gene that transcribes for our friend Titan, which in humans, at least, occurs on chromosome 2. Now, each transcription unit has a sequence just above it on the strand, and that's called upstream. Biologists call that upstream on the strand, and that sequence sort of defines when the transcription unit is going to begin. This special sequence is the promoter, and it almost always contains a sequence of 2 of the 4 nitrogenous bases that we talked about in our last episode: adenine, thymine, cytosine, and guanine. Specifically, the promoter is a really simple repetition. We got thymine, adenine, thymine, adenine, and then A, and then on the other side, AT. Because you know how this works, right? This is called the Tata box. It's nearly universal, and it helps our enzyme figure out where to bind to the strand. Now you'll remember from our episode about DNA structure that DNA strands run in one of 2 directions, depending on which end of the strand is free and which end has a phosphate bond. One direction is 5' to 3', and the other direction is 3' to 5'. In this case, upstream means toward the 3' end, and downstream means toward the 5' end. So the first enzyme in this process is RNA polymerase, and it copies the DNA sequence downstream of the ta-ta box. That's towards the 5' end, and copies it into a similar type of language, messenger RNA. Quick aside, so you'll notice that to read the DNA in order to make enzymes, we need an enzyme in the first place. So it kind of gets chicken egg here. We need the enzyme to make the DNA and the DNA to make the enzyme. So where did RNA polymerase come from in the first place if we haven't made it yet? What an excellent question. It turns out that all of these basic necessities get handed down from your mom. She packed quite a lot more into her egg than just her DNA, so, we had a healthy start. So, thanks, Mom. So the RNA polymerase binds to the DNA at that ta-ta box and begins to unzip the double helix. Working along the DNA chain, the enzyme reads the nitrogenous bases, those are the letters, and helps the RNA version of those nitrogenous bases floating around in the nucleus to find their match. Now, you might also recall from our previous episodes that nitrogenous bases only have one counterpart that they can bond with. But RNA, which is the pink one here, doesn't have thymine like DNA does, which is the green and the blue. Instead, it has uracil. So U appears here in T's place as a partner to adenine. As it moves, the RNA polymerase rezips the DNA behind it and lets our new strand of messenger RNA peel away. Eventually, the RNA polymerase reaches another sequence downstream called a termination signal that triggers it to pull off. Now, some finishing touches before this info can safely leave the lair. First, a special type of guanine is added to the 5' end. That's the first part of the mRNA that we copied, and that's called the 5' cap. On the other end, it looks like I fell asleep with my finger on the A key of my keyboard, but another enzyme added about 250 adenines onto the 3' end. This is called our poly A tail. These caps on either end of the mRNA package make it easier for the mRNA to leave the nucleus. They also help protect it from degradations from nearby passing enzymes, while also making it easier to connect with other organelles later on. But that's still not the end of it. As if to try to confuse me to protect the secret Hot Pocket recipe, the original recipe book also contains lots of extra misleading information. So just before leaving the nucleus, that extra information gets cut out of the RNA in a process called RNA splicing. And it's something like editing this video. This process is really complicated, but I just had to tell you about two of the key players because they have such cool names. One, the SNRPs, which are small nuclear ribonucleoproteins. These are a combination of RNA and proteins, and they recognize the sequences that signal the start and end of the areas to be spliced. SNRPs bunch together with a bunch of other proteins to form the spliceosome, which is what does the actual editing, as it were, breaking the junk segments down so their nitrogenous bases can be reused in DNA or RNA, and sticking together the two ends of the good stuff. That good stuff that gets spliced together, by the way, are called the exons because they will eventually be expressed. The junk that gets cut out are just the intervening segments or the introns. The material in the introns will stay in the nucleus and get recycled. So, for instance, Titan down there is thought to have hundreds of exons when it's all said and done, probably more than 360, which may be more than any other protein. And it also contains the longest intron in humans, some 17,000 base pairs long. Man, Titan, it is just a world record holder. So now that it's been protected and refined, the messenger RNA can now move out of the nucleus. Okay, so a quick review of our Hot Pocket Mission Impossible caper so far. We broke into the layer containing the instructions, we copied down those instructions in shorthand, we added some protective coatings, and then we cut out some extra notes that we didn't need, and then we escaped back out of the lair. Now I have to actually read the notes, make the machinery, and assemble the ingredients. This process is called translation. So next, rewind your memory or just watch that video again to the episode about animal cells. Do you remember the rough endoplasmic reticulum? I hope you do. Those little dots on the membranes are the ribosomes, and the processed messenger RNA gets fed into a ribosome like a dollar bill into a vending machine. Ribosomes are a mixture of protein and a second kind of RNA called ribosomal RNA, or rRNA. And they act together as a sort of workspace. rRNA doesn't contribute any genetic information to the process. Instead, it has binding sites that allow the incoming mRNA to interact with another special type of RNA, the third in this caper, called transfer RNA, or tRNA. And tRNA really might as well be called translation RNA because that's what it does. It translates from the language of nucleotides into the language of amino acids and proteins. On one end of the tRNA is an amino acid. On the other end is a specific sequence of 3 nitrogenous bases. These two ends are kind of matched to each other. Each of the 20 amino acids that we have in our body has its own sequence at the end. So if the tRNA has the amino acid methionine on one end, for instance, it can have UAC as the nucleotide sequence on the other end. Now it's just like building a puzzle. The mRNA slides through the ribosome reads the mRNA 3 letters at a time, each set called a triplet codon. The ribosome then finds the matching piece of the puzzle, a tRNA with 3 bases that will pair with the codon sequence. That end of the tRNA, by the way, is called the anticodon. Sorry for all the terminology. You need to know it! And of course, by bringing in the matching tRNA, the ribosome is also bringing in whatever amino acid is on that tRNA. Okay, so starting at the 5' end of the mRNA that's fed into the ribosome, after the 5' cap, for almost every gene, you find the nucleotide sequence AUG on the mRNA. The ribosome finds a tRNA with the anticodon UAC, and on the other end of that tRNA is methionine. The mRNA, like a mile-long dollar bill, keeps sliding into the ribosome so that the next codon can be read, and another tRNA molecule with the right anticodon binds on. If the codon is UUA, then the matching tRNA has an AAU on one end and a leucine on the other. And if the mRNA has an AGA, then the matching tRNA has a UCU on one end and an arginine on the other. In each case, that new amino acid gets connected onto the previous amino acid, starting a polypeptide chain, which is the beginning, the very beginning of a protein. But it turns out that there are lots of different ways to read this code, because UUA is not the only triplet that codes for leucine. UUG does too, and arginine is coded for by 6 different triplets. This is actually a good thing. It means we can make a few errors in copying and transcribing and translating DNA, and we won't necessarily change the end product. The process continues, with the mRNA sliding in a bit more, and the ribosome bringing in another tRNA with another amino acid, and that amino acid binding to the existing chain, and on and on and on and on and on. Sometimes for thousands of amino acids to make a single polypeptide chain, for example. This whole word is basically just the names of the amino acids in the sequence and the order in which they occur in the protein, all 34,350 of them. But before we can make our own Hot Pockets, and that string of amino acids becomes my muscle tissue, we have some folding to do. That's because proteins, in addition to being hella big, can also contort into very complex and downright lovely formations. One key to understanding how a protein works is to understand how it folds, and scientists have been working for decades on computer programs to try and figure out protein folding. Now, the actual sequence of amino acids in a polypeptide, what you see scrolling along down there, is called its primary structure. One amino acid covalently bonded to another, and that one to another, and to another in a single file. But some amino acids don't like to just hold hands with 2 others. They're a bit more promiscuous than that. The hydrogens on the main backbone of the amino acids like to sometimes form bonds on the side, hydrogen bonds, to the oxygens on amino acids a few doors down. When they do that, depending on the primary structure, they bend and fold and twist into a chain of spirals called a helix. We sometimes also find several kink strands laying parallel to one another called pleated sheets. All those hydrogen bonds and pleated sheets are what make silk strong, for instance. So in the end, our promiscuous amino acids lead to wrinkled sheets. These hydrogen bonds are what help give these polypeptides their secondary structure. But it doesn't end there. Remember the R groups that define each amino acid? Well, some of them are hydrophobic. And since the protein is in the cell, which is mostly water, all of those hydrophobic groups try to hide from the water by huddling together. And that can bend up the chain some more. Other R groups are hydrophilic, which, if nothing else, means that they like to form hydrogen bonds with other hydrophilic R groups. So we get more bonding and more bending, and our single file line has now taken on a massively complex 3-dimensional shape. It also explains why I can fix my bed head by wetting my hair with water. The water helps break some of those hydrogen bonds in the keratin, which relaxes its structure. That way I can comb it out, and when it dries, those bonds reform, and voila, perfect hair. All of this shape caused by bonding between R groups gives our polypeptide its tertiary structure. So now we have a massively contorted polypeptide chain, and it actually contorts very precisely. Sometimes just one chain is what makes up the whole enzyme or protein. In other proteins, like hemoglobin, several different chains come together to form a quaternary structure. So quick review of structure. The sequence is the primary structure. The backbone of hydrogen bonds forming sheets and spirals are the secondary structure. R group bonds are tertiary, and the arrangement of multiple proteins together give the quaternary structure. These polypeptides are either structural proteins, like this thing at the bottom here, that you can find in your muscle, or in my Hot Pocket. They might also be enzymes. Enzymes like do stuff. They can cut up biological molecules like I do with this chef's knife. They can mix stuff, and they can put stuff together. So from that one recipe book, we get all the ingredients and all the tools necessary to make me, which is better than a Hot Pocket. Would you all agree? Now take your time with this stuff. Feel free to watch the episode a couple of times, because next week we're going to talk about how cells swap all of this genetic information through reproduction. Thank you for watching this episode. By now, you should probably know how this works. You can click on any of the links over there and it'll take you back to that point in the show as long as you're not watching on your cell phone. It doesn't work on cell phones. I apologize for that. Thank you to everyone who helped us put this show together, and thank you to you for watching it today. If you have any questions about this episode, please leave them in the comments below, or you can get us on Facebook or Twitter. And that's all. Goodbye.